Hi everyone. Today we are going to study a beautiful theme of biology, and that theme is mm, gene X, genetic information. Genetic information. I was going to choose which color should I write, and I forgot to teach what the theme which I'm going to teach. Okay, genetic information. Okay, so all the life processes, right? They involve the expression and transmission of genetic information. I'll give you simple example of day-to-day life. Y- Life process. Suppose you ate potatoes, paneer, whatever you ate. I don't know what you ate. Right now or few minutes ago, long time ago, whatever you ate, that food needed to be digested, right? So the food needs to be digested, and for that you need enzymes. Enzymes are proteins. So enzymes are proteins, and for that enzyme to be made, that protein to be made, you need the order from the cell. The order has came that the food is there, we need to digest, we need enzymes, and that has to be made from the information carried in your DNA. So DNA carries the information. DNA has particular gene for the synthesis of that particular enzyme. So see here, this is the gene carried in the DNA, right? And then from the DNA we will get the protein by the process of gene expression, which we will study after some time, not some time, in the next lecture, right? That how from a gene we get protein. So, but the simple thing is that gene got expressed and the protein was made. See, inside a organism you have cells, right? Inside you have different cells, and different cells have DNA in the form of chromatin. When the cell is not dividing, when the cell is going to divide, the DNA will condense, this chromatin will condense, and you will get chromosome that will replicate. DNA replication will be there, right? And then the cell will divide, and that genetic information genetic information inside the DNA will be transmitted to the daughter cells, from one cell to the two cell. For example, a child is running on the road. The child fell down, and simply the skin peeled in a little part of the knee, right? That little injury is healed within few days. So that healing needed cell division, and the skin cells in your skin, they rapidly divided and the injury got healed. So from one cell, multiple cells you got there to heal the injury, and the DNA in the cell, in one cell, was transmitted to the different cells. So that was also a process of life, healing, right? These simple things, digestion, healing, all these things, right, they involve the genetic information. Okay, if we say, why inside a non-dividing cell the DNA is scattered loose in the form of chromatin? Why it is not tightly packed up? See, when the DNA is loose, it is very easy for the enzymes involved in gene expression. That is, from when you have to make protein, then from DNA you need to make RNA, from RNA you need to make protein, and for that you need different enzymes and all, and they can easily reach the gene in the DNA when the DNA is loose, when it is in the form of chromatin. When it will be highly coiled, then how the things will enter? Suppose I say there is a thread, and there is a beautiful portion I have colored here, suppose, and I ask you, okay, you go there and reach it. So if the thread is loose, you can easily touch that portion. But if I highly coil the thread and make a roll of that, it will be tough for you. Just like that, when the gene has to be expressed, the DNA has to be loose. So DNA gets compacted in the form of chromosomes only when the cell is preparing for cell division, and DNA is colorless. So if you have to see the DNA, you need to color. See here, the garlic root cells have been studied for cell division, and we have studied the DNA here, and the dyes used are different. For example, orcine ethanoic stain here toluidine blue stain. See, you don't need the name of stains. I'm simply saying that the dye, different dye imparts different color. Here blue color, here purple color. So different dyes impart different color to the DNA, and then you can see the DNA because of the dye in a cell that is going to prepare, that is undergoing cell division. See here, you can see the chromosomes here in different phases of cell cycle that we will study when we will study the cell cycle. Okay, now if I say DNA, the genetic material, that DNA is a double helical structure. You know, this structure was studied by J.D. Watson and Francis Crick and all, we will study later. This double helix is actually made up of two chains made up of nucleotides. So if I say, what are the building blocks, chemical building blocks of DNA? Nucleotides. So what a nucleotide is? A nucleotide is made up of a nitrogenous, that is nitrogen containing base, which are of different types: adenine, thymine, cytosine, guanine. And if RNA is there, uracil will be there. But right now we are studying DNA. Okay. A pentose sugar in DNA, it is deoxyribose. If I say RNA, it is ribose. And a phosphate group. So these three things together make up the nucleotide, and these nucleotides are arranged in sequence, in a particular sequence, and you get the DNA. And the two strands are bonded together, right? Okay, we study hydrogen bonds and all, later we will study. Okay, now if I say, what is the abbreviation of the nitrogenous bases? ATG, ATCG, right? Adenine, guanine, cytosine, thymine. And the abbreviation for nucleotide is also same, ATGC, right? Okay. Let me give you the example of one nucleotide. See, what is this nucleotide? DAMP, deoxyadenosine monophosphate. But we easily simply say A. It is more easy, right? Okay. How to keep the names of nucleotides and all, we will study when we will study DNA. Okay. Now the nucleotides arranged in a particular sequence, and that particular sequence is the one that is responsible for different genes, which are the units of inheritance. So a gene that codes for a particular enzyme will not code for antibody, right? So how the genes are different? Simply because of the sequence in which the nucleotides are arranged. For example, there are three alphabets R, A, T. If I arrange like this, it becomes rat. If I say TAR, it becomes star. If I say ART, it becomes art. The meanings are different. Just like that, these four nucleotides are arranged in different sequences, and you get different hundreds and thousands of what? Genes. Okay. One day you were one-celled organism, zygote. There was true friendship between the sperm and the secondary oocyte. See, this is not ovum, this is secondary oocyte, which is fertilized by the sperms. That thing we will study when we will study reproduction. So male female gametes fused and you got zygote. So you were zygote, but from that one single cell you became, this much came out of your mother's womb, just like my daughter came out of me and I came out of my mother, right? Okay. So, and then the baby grows, develops and becomes big. How all these things happened? All these things developed according to the information carried in the DNA, genetic information carried in DNA. Deoxyribonucleic acid. See, if I say this single cell, from this single cell side, not this single cell, yeah, this fertilized cell, the sperms and all will go, this will be one fertilized cell here. See, this single cell zygote, this is having the information to make the entire human being. This cell divides, keep on transmitting, that is the transmission of DNA will be there, right, in this life process of reproduction, in this, right, in the process in which the one celled organism is going to be converted into a big organism, full organism, bigger, here it will be small organism only, right, and then one day that organism will be big because of different cell divisions. One more thing, when you grow and develop, it's not just cell division, it is cell division plus differentiation. See, right now you have trillions of cells in your body, and the cells are totally different. A nerve cell is different from the cell inside your retina. Right now you will say, ma'am, we have nerve cells in retina too. See, in the retina also there you have different types of cells, right? Okay. If I say a cell of skin, that is different from the cell of liver. A cell of kidney is different from the cell of islands. So how these different types of cells made from the single zygote? There was differentiation when the baby was developing inside the womb. There was cell differentiation, and from same types of cells we got different types of cells, and that differentiation was also a part of development. And now in your body, from the skin cells you will get only the skin cells. If that skin cell will divide, you will not get any other cell. If the liver cell divides, you will get that particular liver cell only. If the cells inside your eye lens divide, you will get the lens fibers only, those lens cells only. So there was differentiation during the development, and that cell's identity was formed, right, and each cell has different function. Okay, now in the next lecture we will study gene expression. Thank you. Bye.